Hi, everyone. My name is Frank Wendt. I am a genetic epidemiologist in the Division of Human Genetics in the Department of Psychiatry at the Yale School of Medicine. And today I'll be presenting some data that my collaborators and I have put together, aiming to leverage bioinformatic resources to better tackle challenging biological substrates and DNA profiles. I don't have any conflicts of interest to disclose, and I have no relevant funding for the data presented today. A very brief overview. I am going to give a little bit of a background on some of the concepts in the forensic genetics and genomics community before I dive into bullet points 2 and 3, which are some data that we've been putting together for 2 projects. And then we'll wrap up with just some next steps and big picture information. So diving into some of the background here, this should be fairly familiar for everyone, but in the presence of a relatively diverse audience, we will go through some basics. Humans have 22 autosomes, 22 pairs of autosomes. One of each chromosome is inherited from both parents, and we also have 1 pair of sex chromosomes that are shown on the bottom right of that yellow square, the X and Y chromosomes, the presence of a Y chromosome, dictating the biological male sex. Within the human body, chromosomes are contained in cells, which are contained in organs, which are all over our body. And if we go sort of the other direction and looking really closely at what's within a chromosome, we see that this oblong structure can unwind and unwind and unwind. And we ultimately end up at this string of A's, T's, G's, and C's that are contained within our DNA. And this will be the focus of our talk today. These A's, T's, G's, and C's tell our cellular machinery how to make the appropriate pieces of what goes on in our body, what those pieces are supposed to do, and when those pieces should stop doing what they're supposed to do. So in the forensic science community, we focus on primarily 2 types of genetic variation for identifying individuals. The first is the short tandem repeat, shown here on the top of the slide. In blue and purple, I'm showing the pieces of DNA that could be inherited from the maternal or paternal side of your background. From the paternal side, we see that this individual received 3 copies of that GATA sequence. From the maternal side, we see that this person received 4 copies of that GATA sequence. At this STR locus, this person would be considered a 3-4, indicating the length of those alleles at the locus. For single nucleotide polymorphisms, we see a much different structure here. In red, I'm highlighting this SNP motif. We see that from the maternal side, this individual received the G allele, and from the paternal side, this person received the T allele. So both of these things are used in forensic science, and both of them have some benefits and some drawbacks. STRs are highly variable. They're really good at distinguishing people from everybody else. We don't need a lot of them. But because they are so large and because they are repetitive, they can have some problems with their amenability to the chemistry that we use to genotype these types of loci. SNPs, on the other hand, are less variable. They are still pretty good at distinguishing 2 people, but we need many more of them to do so. And they don't suffer from the same chemistry issues that we have with STRs. So in certain circumstances, these could be really useful choices to pick an STR versus a SNP based on your question. So in the forensic science community, STRs are kind of considered the gold standard, especially for casework. We're seeing here on the bottom right of the screen an electropherogram derived from PCR-CE approaches. If I zoom in on one of these positions, we see that the electropherogram here shows at one locus the 3-4 allele designations that we see in the person on the top of the screen. The heights of those 2 triangles or peaks are a little bit different from one another. And that reflects different relative fluorescence units derived from the capillary electrophoretic procedure that these go through. We can also have an instance where we observe several alleles at a locus. So in this example, I'm now showing you that same locus, but we have 4 triangles or 4 peaks. So is this a 1-person profile? Is this a 2-person profile? Is this a 3-person profile? We can occasionally, with some additional information, say, hey, we found a match. It's a 2-person mixture. Our victim is a 4-5, so they carry 4 copies and 5 copies of that STR. Our person of interest is a 3-6. But what does that actually mean? What do we do with this information? We can perform some statistics that rely heavily on population genetics. On the right side of the screen, I'm showing the distribution of allele frequencies for this hypothetical locus in 3 United States population groups: European American in green, African American in yellow, and Asian American in orange. We can use this information to calculate the rarity of observing the person of interest's alleles at this locus. And typically, all of these combined estimates in the table here would be work report related to this profile. So I'm not going to touch on the chemistry of massively parallel sequencing, but I do want to introduce the power that this has, especially in terms of mixture deconvolution. So on the left side of the screen, we see sort of the same scenario I've been showing on the last couple of slides. We have a couple of alleles at a locus. By length, we may make some assumptions about whether or not this is a 2-person, 3-person, 1-person mixture. If I gave you some more context and said that the victim is a 3-4, you might say, okay, the other contributor or contributors might be a 4-6 or even a 4-4. But what if I gave you more context and said that there's only one other contributor? You may then say, okay, we can make some pretty confident assumptions that this is a 3-4 mixed with a 4-6. But massively parallel sequencing lets us take this a little bit further and say, I can actually tell you what part of that 4 peak came from our victim and what part of that 4 peak came from our person of interest. And this is done using the sequence level information that is contained in each of those triangles. So we can break apart that 4 triangle into 2 pieces, the red part that may belong to the victim, for example, and the blue part that may belong to our person of interest. So now moving into some of the actual studies that we're doing in the context of mixtures and some of these more complex DNA profiles. So first I want to introduce the sort of rationale behind why we have started predicting STR genotypes from identity informative SNPs. So I've presented both of these types of loci on the last couple of slides. We know that STRs are highly polymorphic. We know that they have larger amplicon sizes, but they do tend to suffer from PCR artifacts. And this makes them especially difficult to really get good information from in the context of a compromised substrate such as human remains, or a substrate that may be heavily environmentally insulted from something like humic acids. On the contrary, the SNPs are smaller amplicons, so degradation may not be as much of an issue. They don't have PCR artifacts, so untangling stutter from true peaks may not be a problem. They are also less polymorphic, so we do need many more of them. But they are easier to type in compromised substrates. So much of the approach to tackling sort of challenging human remains or challenging environmentally assaulted samples has been from the side of the chemistry. We've taken a different approach and said, what if we just use the data that we can get from the identity informative SNPs and better use the data that we can get from this profile? So to do this, we rely on a process called imputation, and this is basically a statistical inference of unobserved genotypes at the STR loci of interest using identity informative SNP loci of interest. So this relies heavily on the concept of linkage disequilibrium between SNPs and STRs. In a diagram on the right side of the screen, I'm showing sort of roughly how this works. If we have our study samples that contain our SNP haplotype of interest, we also have a set of reference information that includes typically whole genome sequenced reference panels that include our STRs in the black box and our SNPs of interest all in the same read. And then we can basically perform a VLOOKUP, or look up our haplotype in our reference and say, okay, this matches, and then I can put a weight to how well it matches and what I think the gaps are in the STR locus. So we performed this process in a couple ways, and I'll show that on the next 2 slides. But first, we identified STRs that were in high linkage disequilibrium with SNPs of interest from the forensic DNA signature prep kit. We evaluated their heterozygosities. We compared some random match probability information, and then we're also investigating a leave one out analysis to investigate how some incompleteness of your II-SNP profile may actually contribute to the data that you can receive in forms of STR genotypes. So to very quickly summarize why we're doing this, STRs likely won't work well in the context of compromised human remains. SNPs could work a little bit better. Partial SNP profiles may actually, in our study, be predictive of partial STR profiles, such that the random match probability could become a little bit better. And then ultimately, the long term here is to test whether or not what we're doing in the computer actually reflects what happens in real life. So here is a study overview. We took two sort of sets of SNPs. The first was an entire SNP array's information of several million SNPs. We also then reduced that SNP count to only the 94 identity informative SNPs from the forensic DNA signature prep kit. We performed SNPster imputation, and then we compared the heterozygosities and allele counts for three sets of STRs: the CODIS loci, the Novroski et al. 73-plex, and the highly concordant STRs that we identified through this procedure. So this work has primarily been spearheaded by Angelica, who's a master's student in Nicole Novroski's lab at the University of Toronto. She took these 222 SNPs, which represents the 94 II-SNP amplicons. We received just under 170,000 STRs imputed. 67,000 of those had global heterozygosities greater than 0, and 24,000 of those had heterozygosities greater than 25% in three major global population groups of interest. We did use the 1,000 Genomes project for this, and we asked two primary questions: what's going on with CODIS and the Novroski et al. 73-plex, and then what's going on with the other loci that we found that were actually imputed really well. So first, under ideal conditions, so when we gave the procedure the entire SNP array's worth of information, the CODIS loci and the Novroski et al. 73-plex generally imputed pretty well. So when we compared this to the imputation using just the II SNPs, we only identified three STRs that actually gave us some meaningful information in terms of imputation success, in terms of concordance with the heterozygosity and allele spread with the truth data. These include one of the Novroski et al. Loci, and then two of the CODIS loci listed here. What's more interesting is that we actually found 545 STRs that were well imputed using our 94 II SNPs. I'm showing here the heterozygosities of those STRs in three of our population groups using the II SNP and the SNP array information. Those are pretty similar, really nice overlap, and the allele spreads are actually perfect between the two of them. So our next steps, because the heterozygosities of these loci were quite low, they're probably not super useful for HID, so human identity purposes. So we are currently characterizing their ancestry effects as well as some of the phenotypic consequences of these 545 STRs. This manuscript is in preparation, with Angelica taking the lead under the mentorship of Nicole Novroski. So next we'll look at simulating mixtures. Very briefly, what does it mean to simulate data? We actually do all of this on the computer, so we can take real-world scenarios and then make data out of these real-world scenarios. The value of doing this is that we have a lot of control over what conditions affect the data that we produce. This is both time and cost effective and scales to very large sample sizes, which is really, really great for early career investigators like myself. One of the major limitations to this that I will be super transparent about throughout the presentation is whether or not there is some sampling error or stochasticity in how these data are simulated. So I'm showing here also a picture of an undergraduate student, Ashley, in Nicole Novroski's lab. She has been instrumental in putting all of this work together, so I do also want to make sure that she gets the shout out that she deserves. So we used the R package Forensim that was developed as a set of tools to study and interpret DNA mixtures. We used this package to simulate genotypes and mixtures and population data based on published allele frequencies. The package also allows you to perform some statistical interpretation of the data that you generate. We'll be focusing on the likelihood ratios, but there are a whole bunch of other statistics that you can generate in Forensim. So our overall goal was to assess the reproducibility and amenability of Forensim to large population genetic and mixture studies. We relied on the population allele frequency data from Novroski et al. In 2016, using African American, Asian American, European American, and Hispanic population groups, all of which have been genotyped for STRs in the Forensic DNA signature prep kit. Our study design included generating 50 population replicates of each of our target populations for a total of 38,900 individuals. We performed a couple of population genetic parameters on these 50 replicates and then compared those back to the Nicole Novroski estimates. We then took all of the individuals that we were studying and created 2,400 same ancestry mixtures. What that means is that we took two people of African American from the African American cohort, mixed them together, and then performed our mixture match statistics using those individuals. So our primary endpoint here was the likelihood ratios, and we did compare those back to the same likelihood ratio, the likelihood ratio for the same profile calculated with Novroski et al. And I do want to point out here that for simplicity, all of our likelihood ratios assume the most basic numerator and denominator. So we essentially said that all of the alleles observed in the profile can be explained by this person versus cannot be explained by that person. So in the context of a more complicated or several alternative hypotheses, much of the data that I present today may not hold. So looking at our population genetic information, we're showing here in panel A the distribution of Hardy-Weinberg equilibrium p-values across our 4 American population groups. We see the vertical dashed line indicates the nominally significant p-value of 0.05. So everything to the left of that line indicates a significant deviation from Hardy-Weinberg equilibrium, at least at the nominally significant level. We tabulate what those deviations look like across all of the STRs in the forensic panel in object B here. We see that there is really no indication that a specific locus is causing a problem here. And actually, when we perform our multiple testing correction, we still see that this is not really any indicator that a specific STR is giving us issues in terms of our simulated population data. Unfortunately, our pattern of significant deviations from Hardy-Weinberg equilibrium also look very different from what Novroski et al. Observed in the 2016 article. In terms of allele frequencies, I'm showing here in black the point estimates in the 95% confidence intervals from Novroski et al. In red, blue, purple, and green, I'm showing the point estimates from our 50 simulated populations. You'll see that all of those distributions do fall within the 95% confidence intervals of Novroski et al. And this was initially really exciting to us, but as we thought about it a little bit more, we became curious about how these even minor changes in allele frequency distribution could impact the resulting match statistics for either single source profiles or a mixed DNA profile. So we went on to test how that may affect what our mixture statistics look like. So here we have our simulated mixture results for 2-person, 3-person, and 4-person mixtures. Again, we are only using same ancestry combinations of individuals. So in the top left there, that AFA box, we are only looking at 2-person mixtures, both of whom are of African American, or both of whom are from our African American cohort. We took that mixture and we calculated likelihood ratios for all 200 of the mixtures we created. And those likelihood ratios were derived using allele frequencies from all 4 populations. And what we're showing here is that in every instance, the true contributor population is the one producing the least prejudicial likelihood ratios. And this is a really good sign. This was more of a sanity check for us to make sure that the mixtures we created and the populations we generated are giving us estimates that we expect to see in the real world. I will point out that as we move into that Hispanic column, we do see that a relatively large proportion of the non-contributor likelihood ratios fall below that diagonal dashed line, indicating that the least prejudicial estimate of the likelihood ratio actually came from a non-contributor population group. And I think that this ultimately has some pretty major implications for mixtures, including highly admixed population groups. So here we're showing how your choice of population replicate influences the distribution of likelihood ratios across all 200 of the mixtures that we created per facet or per group of individuals. So on the x-axis, we're showing the standardized mean difference, and this is really just a way to better capture the magnitude of effect size between two distributions, given the standard deviation across everybody from those two groups. On the y-axis, we're looking at the minus log 10 p-value for the FDR adjusted p-value. So the higher the data point, the more significant the difference between our distribution of likelihood ratios and Nicole Novroski's distribution of likelihood ratios. So what you'll notice here is that first and foremost, there are a lot of data points in all four of those facets that are above the dashed line. This indicates that we saw a ton of significant differences when compared to the distributions from Novroski et al. I do also want to point out here that with the exception of the Asian American population, over half of all of the other population groups produced a significantly different likelihood ratio distribution, which is suggesting to us that your choice of replicate and even just using one replicate may not be very sufficient in terms of accurately modeling what you think should happen with the data that you've truly genotyped. I do also want to point out here that in the EUA facet, that's the European Americans, we see three populations that we simulated that actually had extremely large effect sizes, or standardized mean differences, sorry, and extremely significant p-values. So all of these negative data points on the x-axis indicate that our simulated populations produced larger log 10 likelihood ratios. So the profile was actually indicated as being more rare than it would have been in the Novroski et al. Estimate. So that's fine, but do those results suggest that maybe we have an issue with just a couple of mixtures that went into the analyses? So here we're showing how the number of replicate populations chosen influences the likelihood ratio distributions per mixture. So each data point here represents a mixture. Those are shown on the left side of each violin plot, whereas the actual distribution of data is shown on the right side of each violin plot. On the x-axis, we're showing the number of replicates that was included in the calculation of the mean likelihood ratio. On the y-axis, we're showing the difference between that mean and the Novroski et al. Estimate. As we increase the number of replicates, this is unsurprising that we sort of reduce the distribution of mean differences. Thankfully, it's being reduced to hovering around 0, suggesting that there is little to no difference between the replicated populations and the Novroski et al. Populations. However, I'm only showing the AFA subset here just to make the point more clear. After multiple testing correction, we identify that this choice of 10 replicate populations going into your mixture study is where we seem to converge on the narrowest distribution relative to the original distribution. So the means from 10 replicates onward tend to hover around 0, and those distributions have the smallest variance relative to just picking one replicate. So you'll see here that the actual distribution of differences in that one replicate violin plot are not super large. So for two-person mixtures, this may not be super meaningful to include a bunch of replicates, although we still think that statistically this is a significant observation. When we look at the, I believe these are the three-person mixtures, we see a very different story. We are still identifying 10 replicates as being a really good spot for us to target in terms of how many times should we ascertain this data. However, what we're not seeing is that narrowing of the distribution. All of our means do tend to reduce closer to 0, but we still have a substantial number of individuals on the extreme ends of our distribution, such that the variance does not change among these observations. So in some instances, so maybe these 3- and 4-person mixtures, we have not actually identified an appropriate number of replicates yet with which to sort of get a meaningful, useful, and high confidence estimate of what's actually going on. So to summarize here, when using modeling tools, it's really important to pay attention to some comprehensive assessments of the reliability of those tools. And really the impetus behind us doing this project is that we didn't feel like we had literature for that to support our use of Forensim in some of these projects that we want to do. So here we demonstrated that the Forensim data works really well, or sorry, the Forensim package works really well with the Novroski et al. Data. It is fast, it is efficient, we can look at really large sample sizes, but we have not demonstrated that our observations that I presented to you today can generalize outside of the Novroski et al. Data. So this is sort of a natural next step for what we're currently working on. We do want to note that the allele frequency range was quite broad, even though this was contained within the 95% confidence interval of our point estimates from Novroski et al. We did identify a number of Hardy-Weinberg equilibrium deviations, and we think in combination this could indicate that there may be some residual population stratification that is being induced using the Forensim procedure, and this is likely a result of us simulating the exact sample size of Novroski et al. In the event that there are rare alleles in the Novroski et al. Dataset, if we are simulating the same dataset size, we can only sample some of those rare alleles so many times, and if they are not in the right combination, we may observe very different Hardy-Weinberg equilibrium deviations from what was actually observed with the Novroski et al. Data. So in all of our analyses, using a single simulated population to calculate likelihood ratios was extremely noisy. I think that we can confidently say that one replicate in this type of analysis using simulated data is not sufficient to garner any type of confident estimate in what's going on with that mixture. We tended to highlight a population replicate count of 10, although this may be able to go down a little bit if we considered a minor allele frequency adjustment, which we did not do in our study. So it is very common to apply this 5/2n threshold or adjustment to alleles observed fewer than X number of times. And while this may have reduced our population replicate count, our data did not tend to support that low frequency alleles or profiles or mixtures that carry low frequency alleles were the origin of the large distribution in likelihood ratio differences that we observed. So given many of the Hardy-Weinberg equilibrium deviations that we observed, many of the Hispanic population non-contributor likelihood ratios that we observed, we have sort of a standing hypothesis that our results have some pretty considerable implications for cross-ancestry mixtures, and these are currently underway, being led by students from Nicole Novroski's lab. So the manuscript describing the data I presented today is currently in review, so I ask you to patiently await the output from that. I do want to give a shout out to both Ashley and Dr. Novroski for their tremendous support and initiative in getting some of this data, really running instead of just walking. So finally, some acknowledgements, again, to Dr. Novroski and her lab at the University of Toronto, who've been super supportive and working really hard at getting some of these projects going. Dr. Hinda Haned, who is the creator of the Forensim R package. Ultimately, we couldn't do the project if the package didn't exist, so hats off to her for putting this out into the world. And finally, Dr. Rory Rolfs and Nico Caberio at SFSU for really enthusiastic discussions over the last year about the social and legal implications of ancestry, race, and ethnicity on mixture interpretation. I do anticipate that many of these discussions will form some hypotheses for some long-term future collaborative efforts. So finally, if there are questions, I do encourage you to reach out to myself or Dr. Novroski. Our contact information is here: Twitter, email, website contacts. Those are all fair game. And finally, I did want to put out the special announcement that I am guest editing a special issue of Genes titled Source Attribution Using Forensic Genomics. The deadline for this is excuse me, October 20th of this year. If you do have questions about submission, feel free to contact myself or any of the editorial staff through the manuscript submission system. We will hopefully get you rocking and rolling as soon as possible. And I appreciate your consideration for submitting to us. And with that, I don't think I can in person take any questions unless it's via email, but absolutely entertain the option for questions through either endeavor. And that is all. I thank you for your attention.